So very good evening, everyone, and very warm welcome to the third week of our Fundamentals of Bioinformatics, this weekly lecture series organized by UDGAM AI and Health Research Center. So over the last two sessions, we have gradually built the foundation of bioinformatics, beginning with its basic concepts and then moving into biological databases, their different types, and the distinctions between primary and secondary databases. Today, we take that learning a step further. We will understand how sequence data can actually be retrieved, compared, and analysis. And it will focus on sequence databases and pairwise sequence analysis. We will look at protein and DNA sequences, DNA sequence databases, and sequence retrieval. So today's session would help bridge gap between the biological databases and the practical analysis of sequence information, an important step for anyone interested in genomics, be it molecular biology or computational biomedical research. So, and once again, we are very pleased to have Navamita Roy with us to guide us through today's session. And let me briefly introduce about her. So, Navamita has completed her MTech from the Department of Biological Science and Engineering, Manit Bhopal, and is currently pursuing a joint PhD at Manit Bhopal and AIIMS Bhopal. Her research involves application of machine learning and computational approaches to biomedical research, particularly development of predictive and classification models using microarray-derived gene expression datasets. She also complements her computational findings with experimental validation using in vitro cell culture models, helping establish a translational connection between computational predictions and laboratory-based medicine. So Navamita, thank you once again for continuing this journey with us. I'm sure today's session will take our participants to a clearer understanding of how biological sequences move from database into meaningful analysis and interpretation. With that, I will hand over the session to you. Thank you.
Thank you, Dr. Utsu, for this warm welcome. I would like to share my screen.
Yeah, it's visible.
Okay. So, good evening and welcome. And today's session is about a single database, that is GenBank, and I would like to justify spending time on one database before we start a gradual and depth understanding. So, almost every piece of computational biology any of you will ever do begins with somebody else's sequence. So suppose you run a blast search on a bacterial isolate. You pull a reference transcript to design a primer. Suppose you download a gene family to build a tree, a phylogenetic tree. Or you like to feed a training set into a deep learning model. In every one of those workflows, the thing you are standing on is a public archive. And for nucleotide data, that archive is the GenBank. So one of its two partner database, like DDBJ and EMBL. So the question I want us to answer is not just like what is GenBank as in a manner of cyclopedia sense. It is what exactly I am standing on and when I use this data, and what I am responsible for when I put data into it. So now we are going to elaborate it, and we're going to end on end-to-end of the topic. So I'm going to start with the description of the definition of the GenBank. So here is just a formal definition that GenBank is a NIH genetic sequence database, an annotated collection of all publicly available DNA sequence. So there should be three claims, three notions that we are going to introduce, that is NIH, all publicly available, and annotated. So gradually we have to understand one by one what it stands for. So NIH, it is a host for GenBank. GenBank began its journey in the year 1982, that growing out of a sequence collection. And it has been built at Los Alamos National Laboratory back then. And since 1992, it moved to NCBI, that is National Center for Biotechnology Information. So this is a 40-plus-year-old institution, and even older than the way we are searching for. So the next term that we have to clarify, to discuss, what does it mean, all publicly available database or archive? So GenBank holds what has been submitted and released into it. It does not hold just sequence sitting on a hard disk in somebody's lab. And importantly for clinicians, it does not hold controlled access human data by a data access code. So all publicly available is doing a real work or a real-time validation, and that is the boundary of the GenBank. That is the boundary of the public records. So what that term annotated means? So this is, I want you to remember that GenBank is not just a dump of FASTA files. The files format that is available in GenBank is known as FASTA. So it's not just a dump of FASTA files. Every record couples the raw string of bases with a structured description what those bases are and which organism they came from, where the genes actually located in the chromosome, and where the coding region has started and going to end, and which publication reported them and who submit them and when, and the time frame. Everything will get extracted from that particular row of sequence so that instruction is available. So that structured annotation is exactly what makes GenBank usable as a training data for so many machine learning models and so many manipulations and computational biology as a source for feature level analysis, and that act as a substrate for every genome browser we are going to have ever handled in future. So this is a public and comprehensive database. That means it holds all the publicly available DNA sequence data. It's already discussed, posted by NCBI. So one of the three partners in the international collaboration. In last session, I have discussed about the international collaboration that is called INSDC. And it is partnering by NCBI and DDBJ and EMBL. I'm going to elaborate it into the next slide also. And the format that is available, that is the structured format, that is flat file and ASN.1 format. So the distribution formats are the called flat file. It should be human readable, which is what we will read in few minutes. And ASN.1, which is a structured, a machine-oriented format. So basically, NCBI uses internally and in this format internally and distributes for bulk consumers. So the full form of ASN.1 is abstract syntax notation. .1 represents the version of that format particularly. So which regulate, actually it is regulate the interdistribution between the platform. And mainly NCBI deployed such type of data formats, the file formats, for storage and retrieval of bulk number of sequences. Now I'm already discussed, this is a part of INSDC. So GenBank does not operate alone. It is one of three members of the International Nucleotide Sequence Database Collaboration. The full form stands for International Nucleotide Sequence Collaboration, INSDC. And the three members are DDBJ, the DNA Data Bank of Japan, and the national, and it is hosted by the National Institute of Genetics in Mishima, Japan. Next, European Nucleotide Archive or ENA at EMBL, EBI, Cambridge, and the GenBank at NCBI, United States. So these three database exchange data on a daily basis. And I want to be very precise about what that means in practice, because it is the single most useful operational fact of this concept of this conglomerate under the INSTC. So suppose you submit once a data, a raw sequence. If you submit a sequence to ENA in Europe, that records is in GenBank within about a day. So how it is possible? So suppose if you submit to GenBank, it appears in DDPJ. The accession number you are going to get or going to assign is distributed across all three databases. So there is no such things that also depositing in ENA for European reviewers you have already done. So all three databases will get your accession number, and automatically your accession number is equally distributed among all three databases. So for us, there should be no redundancy for a particular search. So this is a main structure of INSDC, actually how INSDC is work. And the second point I would like to mention, that is the resilience and the sovereignty. So three geographically and politically distinct institutions hold a synchronized copy of the world's public nucleotide records. So if suppose one goes down for funding, policy, or infrastructure related lag, the other will survive, or other database will act as a buffer or a supplement for a consecutive database. So how this triad of three database actually constituted by the INSDC, the International Nucleotide Sequence Database collaboration. Next, we are going to discuss in this segment, we are just going to discuss about the release notes and the growth statistics. So what exactly it is? So GenBank issues a full release every 2 months. That means 6 in a year. And each release is accompanied by release notes describing the contents, format, and any upcoming changes, and by growth statistics reported separately by the traditional GenBank division and the WGS division. It means the GenBank has 2 divisions. When they are going to disseminate data, they have 2 divisions. One is traditional GenBank division, that is conventional, and for whole genome sequence data, they have a separate division that is called WGS division. So the full issue is released by these 2 divisions, and that is by twice in a month and 6 in a year. So in last session, I've already discussed, I put a number over there, that as of the June 2026 release, the version is 272. The release number is 272. GenBank held on the order of 57.7 trillion bases across 6.5 billion sequence records. And it is gradually picking up in an exponential rate. Because the whole genome shotgun component grew by a further 3.5 trillion bases in same 2 months. Okay. Some important fact that is we are going to discuss that a release that should have been always fixed. and citable snapshot of the database. That analysis that must be reproducible should record the release number, not just the date of the download. Because the like Entrez and the blast service updated continuously between the releases. Now we have to understand, we have to decode this concept, this string of sentence into our conceptions, what is release, and we are going to distinguish between what is release and what is version and what it has, what is the implication of that. So a release actually by contrast, it is just a snapshot or citable snapshot. You could say it is a fixed number. So suppose you want in a paper is like that, sequence were retrieved from the GenBank in March. Rather than they are saying that sequence were retrieved from GenBank release 272.0, that is gonna accessed by the NCBI FTP site. NCBI FTP means file transfer protocol. They contain some releases, and that should have been fixed, because after 2 months that release going to be changed and there is an addition of the new data. And when you are going to retrieve data by using the tool like Entrez or going to perform similarity search blast, that would vary from time to time. Suppose today you are going, this is a month of August, you are going to curate one data, you extract one data or nucleotide sequence using Entrez. After 4 months, it would change. The result is going to be changed completely. So there should be a release or a version. Just that could be a figure, that could be a number is going that need to be provided the single snapshot or a unique identifier at which release actually you are looking. So that's account for the number of reproducibility of an instrument. So if there is a change of the results, but your release or your version is not going to be changed. The version is going to be fixed, and each and every search, there would be a mention of that particular version or particular release. Whether maybe your results is going to be changed after 2 month or 3 month cycles. So that is the real requirement to mention the release to make an accountable reproducibility of the data. So now, if you just make an overview, just take an overview of a GenBank flat file format, and after that, we're just going to see some live examples, live extraction from the original GenBank site. So before that, I would like to give you a short description of the GenBank flat file format. So NCBI provides an annotated sample GenBank record for a particular organism. Here I have mentioned the Saccharomyces cerevisiae gene particularly, and it is over there at the NCBI site. So we are going to perform that today. So it is a standard teaching example, demonstrates most feature of the flat file format in the single record. So what is the basic configuration? It has the header, or you could say the identifier, or which contain definition, accession, version, and keywords. And the source, where the name of the organism and the lineage or the taxonomical lineage, either it is a plant, it is a animal, it is a fungi, so that accounts for the taxonomical records. So next, we are going to have some linked references, that link publication and submission details. Then the important thing, or you can say the core annotation, is integrated with that. It is called the features, and which contain genes, series, and other annotated intervals. So it could be a primary database. GenBank could be a primary database, but they when this kind of features like annotation, genes, CDAs are going to be implemented on that particular database. It is going to be the act like a composite database. So it is a part of NCBI, and from primary database that NCBI is going to take the data, they're going to extract the data. So it contains some annotation. So how this annotation is going to help you to your analysis when we're going to do hands-on, we'll gradually understand that part also. And the origin. Origin means actually the nucleotide sequence. From origin, they have started the sequence. With right-hand side, it is an illustration of a flat file structure. You will see over there. And by hands-on, I'm going to discuss everything, how actually this is work. So there are 4 ways to search and retrieve data actually from the GenBank. So how they? The first is Entrez nucleotide. So what we will do, we search GenBank for a sequence identifiers and annotation through the Entrez text search interface. I'm going to show you today. Next is BLAST. So BLAST is just a tool that is called Basic Local Alignment Searching Tool. Majorly, we are looking sequence similarity between different organisms, different sequence, or better to more precisely to say what different sequence of different sources, how similar they are. We used to perform local alignment search. So this is called BLAST, and this is also an entity of GenBank. So from raw data, we are going to utilize that and we perform BLAST of it. Next, NCBI E-Utilities. For that purpose, you have to have some idea of BioPython, otherwise it's a bit difficult. And today we're not going to discuss it, but I'm just going to put some flavor of it. In future, while we are going to learn some Python script, maybe I'm going to demonstrate how utilizing Python script or available workflow. Programmatically, one can download the data from NCBI. And another file format that is also a machine-based or program-based, that is ASN1 and flat file format. This is available at the FTP server of the NCBI, and you can get over there, you will get that also. You can retrieve that data from part also. So GenBank have some data usage policy. So this database is designed to provide and encourage access within scientific community to be most up-to-date and comprehensive DNA sequence information. So updation is available over there. And NCBI never impose any instruction. There are no restriction from NCBI on the user or the distribution of the GenBank data. It is completely open access and is founding the principle of the resource. And there should be a third-party right may apply, that is the case-sensitive scenario of the GenBank, where some submitters may claim patent, copyright, or other intellectual property rights in all or part of the data they are going to submit. So NCBI is not in a position to assess the validity of such claim and therefore cannot comment on or grant unrestricted permission for the use, copying, or distribution of the GenBank content. So this is a pipeline. You can say it is a pipeline. So how exactly it is working? So from direct submission from individual researcher and another using NCBI submission tool are the most important source of new data for Gen. So there is a portal that is submitted through one's NCBI submission tools over there. Today we are going to see how this submission instrument actually works. So there is a direct submission portal, and after that there will be a there is a part of processing where automated and manual review for data is going to occur, where integrity and quality is maintained by a computational pipeline that is automated, or either automated or by manual. Then the record is subsequently made available to the public, and after that, on rare occasion, data may be removed from the public view if there is any error is anything occur, and that would be intimated by the submitter end. So standard submission. So what type of submission one could do over there? So GenBank accepts mRNA or genomic sequence data directly determine the submitter. Okay. So mRNA or genomic sequence, that is nucleotide sequence generated by submitter denser, and it's requires source organism information. So the submission must identify the organism, the sequence from where actually it is came from. And annotation is provided by the submitter as a part of submission, because it is not a secondary or tertiary database. It is linked with a conjugate database or a tertiary database, but the main base, the core idea came from the primary database. So the annotation is provided by that particular case. We have recall the concept of primary database format, then the annotation is provided by the submitter as a part of the submitter. So now we have discussed what GenBank accepts. Now we're going to discuss what GenBank doesn't accept. So non-contiguous sequence. Non-contiguous sequence should not be accepted by GenBank. Protein sequence with no underlying nucleotide submission. So if you are going to submit protein sequence, it must have some nucleotide submission. And sequence without the physical counterpart, that the consensus sequences. Next, the primer of the sequences. Primer, in sequencing, in the experimental sequencing, where we have to add primer, especially for the identification of the targeted locus. But if you not remove the primer and alone you are going to submit that, it is not going to be submitted. And if you mix data, mix genomic data with mRNA, nucleotide DNA data with mRNA data, so they are not going to be accepted, and sequence shorter than 100 nucleotides, that is not going to be accepted by the GenBank. So mainly the raw sequence reads from the next generation sequencing platform that is going to be deposited in sequence read archive, that is SRA. And after that, it is channelized in different subsection and different sub-part of the GenBank. So we are going to look today how the different sub-part are associated with GenBank. Major dumping is occurring into the SRA, inside the SRA, that is sequence read archive, and after that it is channelized in different segments of the GenBank. So it is contain NGS, WGS, transcriptome data. But the main majority of the dumping up will occur at the sequence read archive, the SRA, and after that, from that part, it is channelized in different subsystems of the GenBank. So this is a specialized submission types. It contains some complete microbial genome, whole genome shotgun, transcriptome shotgun assembly, third-party annotation that is also available to submit over there, targeted locus study, and high throughput genomic. So it has separate segments. I have already discussed about the subsystem from SRA to different, the data is going to channelize in different submission systems like microbial system, which contain only bacterial genome submission and covered by the bacterial genome submission guidelines. And each and every subsystem, they have some guideline, strict rigid guideline that we have to follow. So next is whole genome or shortcut. So read overlap contigs, small contigs and assembly. the name suggests shotgun, so there is small read and overlap context of theirs assemblies from prokaryotic and eukaryotic projects, bioprojects, with or without annotation. It is going to be submitted over there. Transcriptome shotgun assembly, that is transcriptomic context, computationally assembled from primary data already submitted to SRA, sequence read archive. Okay. Third-party annotation. Third-party annotation of genomic or computationally assembled sequence already available in GenBank. There is a position like third-party annotation. A third party could annotate your data that is already available in GenBank. So that is also accepted, and it is a route of specialized submission types. Okay, another is targeted locus study, that is differentiated from the whole genome or any whole study. That is a targeted local study. There should be large scale target sequencing over across 2,500 sequence across a locus, a particular gene location when is going to be the target, and that's said to be conserved any elements of a particular chromosome that if you need to decode them, and you can submit that data through the targeted locus study. And next is high throughput genomics. That is specifically it is a cloning based sequence, usually cosmids, bats. These data is going to be submitted by high throughput genomic data set shoot. So these are the specialist submission types we're going to discuss one by one in coming session. Okay. So this is I already discussed. There is strict entry targeted locus study. A large scale, if you have a bulk amount of sequences greater than like 2,500, the best way to submit through the targeted locus study. So the third party annotation, the condition is that the sequence data must be already presented in GenBank, and the analysis behind the annotation must be appear in a peer reviewed scientific journal. It means when a third party, and so many bioinformatics company, they are used to perform, they used to analyze the data, they used to apply computational pipelines to annotate the data, and after annotation in a publication format, they have to express that data in a publication format in a peer reviewed scientific journal. So this is a new rule that is set by, and it is NCBI, that is effective in 16 June 2020 sequence, that GenBank will no longer accept personal sequence data from a private individual. That is, this includes personal human chromosome, genome, or mitochondrial DNA data. For example, an individual personal health record, health data from a particular laboratory or healthcare provider. Okay. And sequence generated by a direct-to-consumer company for an ancestry or genealogy studies, and because that is very much, these data are very much restrictive, and there is a proper guideline should be followed to disseminate such kind of data to protect the privacy of a particular individual. So these are the case sensitive, and this rule have been imposed by NCBI, policy change rule, since 16 June 2026. These are the confidentiality that should also have been mentioned over here, that authors often worry that the data appearing in GenBank before publication will compromise their work. So GenBank addressed this with a hold mechanism. There will be a hold mechanism. So a release can be withheld. Upon request, GenBank will withhold the release of new submission for a specific period of time. And that is also to show the respect towards the privacy of the researcher or the submitter they are going to expect from NCBI. So publication overrides the hold. If the accession number or sequence data appears in print or online before the specified date, the sequence will be released. So you have an option to tell NCBI when you need to publish. To prevent delay in appearance of published sequence data, authors are urged to report publication as soon as it is available. These are all the privacy obligation policies of human data. No personal sequence data, we already discussed. From 16 June 2026, they are not going to consume the personal sequence data. Okay. So now I have just finished the lecture part or the concept building. Now I'm going to start the hands-on. The first, we need to see how actually the flat file looks like. Just end the PowerPoint show. Just open the browser, and I would like to request you to also follow me with this hands-on. So in browser, you write just GenBank, and it's appeared GenBank overview. Just click on it. So this is a page of main page of the National Library of Medicine, LMM, and this is the GenBank, and already I have selected nucleotide here. This is actually a part of NCBI here, in all 40 database that is attached over here, but I have just type over there nucleotide. I have selected nucleotide. So this is a page, and you click one by one, you will get the option. I'm not going to elaborate, but I'm going to show you what segments it is actually contained, like this. Here you will get the description of the GenBank. You get the submission type, submission tool, update GenBank records. You can also get the search bar over here also, like this. BLAST, and from BLAST you will perform a basic local alignment search. And from statistics, you will see the current statistics. I'm just going to click once and see how it is showing and exponentially increasing. So it has two segments, GenBank traditional and WGS. And you see there is a margin point over there. Sequences and bases. This is a number of bases, this is a number of sequences. Like in billions and billions of data. And now WGS is override. Sequences is override the traditional GenBank. And this is the statistics of the sequences. December 1982, I've already told you about the version. There will be version changes. So it is 6,80,338 in 1982. And there is release of 3 times. And this is a release number. And how many times it has released? There should be a specific number with timestamp. That is called release. There is 3 release in 1982. It has started back then, and now just see the number in the deluge and just scroll it down. And August 2026, and we have started our session, this bioinformatics session. That time it was 272, and now the version is changed 273 now. So August 2026, I think 82. It's billion range data is there. So how it is actually showing, we get the live statistics, and after 2 months it is gonna devised, because again there is an incorporation of data into this stockpile. So from statistics you will get that. So this is sample record, revision history, sequence IDs you will get. Like in a submit option is there. And for the submitter, they use the submission tools. We'll discuss in next session what exactly submission tools are. And it is available. There is submission types also there, submission portal is there. And sequence data processing pipelines also there, how the process is going. So it is genomes, complete genome submission guide for prokaryote, eukaryote, annotation examples. And There is a separate segment for whole genome sequencing. They have maintained, okay, for both prokaryotes, eukaryotes. Okay. There is also metagenomes, and especially highlighted the microbes, metagenomes, targeted sequence analysis or locus specific sequence analysis. This is the INSDC. If you need to learn about INSDC, we'll get over there. And in documentation, you will get everything. The information. And this is a route of specialized submission that is already discussed. TPA, TLS, high throughput, EST, GSS. Those are actually related to the next generation sequence data that they have extracted next generation sequence data and they distributed in this segments: expression tagged sequence, high throughput, targeted local sequence, third party annotation. TPS stands for third party annotation. So these are the segments where it is actually there. Now I'm just going to the main page about GenBank. So there is an annotated sample GenBank record. Okay, we just need to check about it. What exactly this is. So we're going to click on it. Okay. So this is a sample GenBank record. See over here. So how it's actually, this is the flat file looks like. We'll read one by one, but those are the meaning. So first scroll it down and check it. How this is called the flat file format. CDS, chain CDS. I'm going to describe one by one everything. The origin. From origin, you can see there is around 5,000 nucleotide sequence. From origin, it has started. This is small letter initials of DNA sequence. So at the bottom, you will get the field comments. So what exactly, how many fields are there? Maybe there's a 6 fields and respective fields. What is the meaning, definition, what stands for is going to be. So sequence, locus name is there, locus name, sequence length, how long the sequence is. Next, it would be the molecular type, what type, either it is DNA, RNA, genomic RNA, precursor RNA, all the details you will get. And next, GenBank division. This is called the taxonomical division. So this is very important. So, and this part is also linked with the taxonomy browser. So the GenBank division to which record belongs is indicated with a 3-letter abbreviation. These are the 3-letter abbreviation. So these example particularly that the Saccharomyces cerevisiae that comes under PLN, plant algae fungi. So there is 3-letter abbreviation. So first we have to know what exactly 3-letter abbreviation. So according to the taxonomical arrangement of the organism, there are 18 divisions. So there is different type of organisms like PRI. When it is represents primate sequence, that is PRI, and the raw data will comes under the primate sequences that start with PRI, and it is present over there into the flat file format. I'm going to show you. First, I discuss one by one that part that initials the nomenclature, and it would be much more easier to relocate the concepts, and we're going to match and we're going to perform the hands-on file format. So PRI is primate sequence, ROD stands for rodent sequence, MAM other mammalian sequences except primate and rodent, BRT other vertebrate sequences, INV invertebrate sequences, PLN plant, fungal and algal sequences. This is a fungal sequences, Saccharomyces cerevisiae. So it is comes under PLN. And I'm going to show you, you will see the PLN into the flat file. At the beginning, into the locus section, you will get that term, that code, PLN, 3-letter code. BCT for bacterial sequence. If one would like to derive bacterial genome sequence, that comes under BCT. VRL is viral sequences. PHG, bacteriophage sequences. SYN, synthetic sequences. UNA, unannotated sequences. EST, expressed sequence tag, EST sequences. It stands for if you don't have that particular genome sequence or the nucleotide sequence, so there is some expressed sequence tags are there. So this is called EST. Mainly, it's a transcriptome based sequence that provide you some example of expression. From expression, you will identify that particular genomic sequence. Patented sequence, STS sequence, sequence tagged site, GSS, genome service sequences. GSS is also like EST, but when you have bulk sequences are present, that is called GSS. HTG sequences, high throughput sequences. HTCs, unfinished high throughput cDNA sequences, that is cDNA sequences that are coming from mRNA sequences, particular ENV, environmental sample sequence. So these are the segments, actually, distribution segments, or you can say NCBI taxonomy distribution. So under this, according to the source or the bio project from the sequence comes, the annotation have been done like this way in these segments. So these are the division. So there is a raw data already how the dissemination occurred. Just need to recall the concept again. There is a raw data that is submitted into the SRA. Sequence read archive. That is a dump file of FASTA. Now the data is channelized in different divisions. These are all the divisions of genetic. So you get the modification date, the definition, the accession number. Okay. So this is the segment, the accession part. You may get this in reference sec. So reference sec, it is a separate database of NCBI, and that is a conjugate or a tertiary database. So reference sec, what it contains, they are the reference sequence of the GenBank sequence. Means it is non-redundant. There is no repetition of the data. Well organized, properly annotated when the raw sequence are present into the reference sec database, and that is a version of the GenBank database dataset from the sequence present into the GenBank, that is also present into the reference sec database, but it is completely non-redundant and properly processed. So there is accession number. There is a proper coding of accession number. Like when the accession number start with NT, that constructed from the genomic contigs. The contigs are the small sequences, sequence reads. When we are going to perform a sequencing of a large genome, you have to first, cut that sequence into short sequences. And that short read is going to be termed as contigs, and after overlapping that contigs, you are going to construct the entire sequencing pattern. So this NT is nucleotide, actually. It stands for nucleotide. It's come under the constructed genomic contigs. So when the accession number of a particular sequence will start with NM, so you will automatically that set on your mind that this data is for mRNA. When it is started with NP, that represent proteins, okay, and then NC, that is chromosomes, total set of chromosomes, that is called NC. When the data, when that particular accession started with NC, you will see that. So there is a version of every data already. There, year to year, the version is going to be changed. I repeatedly discuss about that. So there will be a version. There is a GI. A GI is very unique thing, that is Gen Info Identifier. It's called the GI actually. So for each and every sequence, don't consider about forget about everything from where organism source, from wherever it is, just forget about everything. Just consider the raw string of raw sequence AATTGC, like this way. But each and every sequence there must be a GI, a specific identifier, that is called Gen Info Identifier. And it is also changed if there is a change in sequence. If you need to change the sequence, that Gen Info Identifier particularly, that is also going to be changed for a particular nucleotide sequence. For a particular protein sequence that is translated from that particular nucleotide, there is also a GI, the Gen Info Identifier. Each and every line of sequence there is a GI, because it is required to maintain the division if there is a change occur into that. Okay, so keywords are there, source are there, from which organism they are going to search. Okay, and organism details, the references, authors, title, journals, PubMed, everything is available. Okay, journal deposited into the PubMed. It is mentioned over here. So this is the direct submission tool. Okay, and there is some features, and we are going to discuss a feature into the flat file format. Okay, source, taxon, CDS. So this is important. Actually, CDS is important. It represents the coding sequence, from where the coding sequence is going to start. So suppose it is written over there 687 to 3158. It means it has started from number 687 and it is going to enter at the 3158. This is your coding sequence. And in this particular example, this is your start of coding sequence and end of coding state sequence. That means it is the open reading frame. From 687, there is a start codon, and T3158, there is a stop code. So there is a closed arrow symbol or less than arrow symbol. It is there, but it means this is the partial sequence. That is, it represents the 5' only. 5' end of the sequence, 5' end of the partial sequence. Okay. So, and next is 3' end. So 3' end, how this is represent this close arrow signal. So the 3' region is present over at the position 5028. So this is for the partial sequences. For 5' represents like this open arrow, and with close arrow they are represent this is a 3' end. So from this symbols, you are going to identify from where the start codon has started and where it is in data, and what is the we are looking the complementary strand direction 5' to 3' or 3' to 5', we will identify. So there is a complement range. So there is a complement range, that is also a concept. So it is mentioned over there, complement element. So when looking a particular protein, better to say particular gene location, we will understand what exactly complement. So gene is present into a complementary strand, the gene location. So the stand always like this 5' to 3' and 3' to 5'. So the complement region is 3' to 5'. So into that 3' to 5' strand, DNA strand, the particular gene is located. So that particular gene product is located. So when we are going to read.
Naumika,
Yes.
Someone is asking that is there difference between GenBank and FASTA format? How can we extract FASTA sequence?
I'm showing. No, there is not. There is no difference. Actually, FASTA is a format, file format. I'm just going to show them how there would be an accession number. There is a greater than sign, and that is an extractable format of the data from the GenBank. That is FASTA. Fast N, fast P. Fast N for nucleotide and fast P for protein. I'm going to show you how it's look like actually. Okay, just let me finish the particular fields, then I'm going to show you. And this is a part of Entrez. So this is a fast tool we are going to use today, that we are going to extract the particular sequence using Entrez. And Entrez is a retrieval system. Better to say it's a retrieval system. We're going to use that, and we will see. This is a what is the format actually? That is called the FASTA format. The sequence is submitted in a FASTA format. I'm going to extract it in FASTA format. There is string of nucleotide sequence. I'm going to show you. Okay, GI translation gene always there. So origin is there. So now just go back to the flat file format, how it's look like. Okay, so there is a locus, a particular segment of the chromosome, SCU 49845. This is called the accession number, and obviously you have mentioned in FASTA format, you will get this accession number also over there. So I request you to all, please note that particular accession number, that is SCU 49485. So SC stands for Saccharomyces cerevisiae, and this is U49485. 49845 is the accession number. The length it has 5028 base pair. It's a DNA sequence, and PLN. And the division, the taxonomic division is PLN, that is plant, alga, funga. And this is a Baker's yeast, Saccharomyces cerevisiae comes under PLN. And 21st June, it has released 1999, long back, 25th June. The timestamp is there. So in definition part, we'll get the name of the organism and the TCP1 beta gene. So we'll get the gene, the particular gene information, the sequence information, that is TCP1. Actually, it is a molecular chaperone protein. It's mainly help to protein folding. And okay, and partial CDS of it, and AXL2P and Rev7 genes. That is a complete CDS available, these two genes. So there is a mention of 3 genes, TCP1 beta, AXL2P, and Rev7P genes. Okay. And partial CDS, the coding region, coding sequence available for TCP1 beta. That is partial means either 5' or 3'. We'll see what type of it is mentioned over there. And AXL2P and Rev7 genes, complete CDS. I mean 5' to 3', 3' to 5', everything is available. Now this is the accession number, U49845. That's going to help you to extract the FASTA file format. Okay. FASTA is just a text-based readable file format. Mentioning the flat file, this FASTA is a type of flat file for nucleotide sequence. Okay, so this is the version name is there, and the genomic identifier is there. Okay, the source name, organism, eukaryota, taxonomy available. Reference basis 1 to 5028. In total 5028. Okay, available. An author's name who have published, and this is cloning and sequence of Rev7, a gene whose function is required for DNA damage induced mutagenesis in Saccharomyces cerevisiae. So this is actually cloning. What type of experiment? Cloning experiment. So already we discussed, while I discussing the PowerPoints, I have mentioned that if you need to submit anything, any data, you have to make a publication in a peer-reviewed journal. After that, you are only allowed to submit that data to the NCBI. Okay. So this is a journal made, reference, authors, title, selection of title of the publication. Okay. PubMed, this is available in PubMed reference title, and this came through the direct submission. If there is a specialized submission, if there is present, it would be mentioned over there. So, but it is comes under direct submission. When it is submitted, it is there also. This is the source. Now from CDS, it is, you can understand this is a partial sequence for particular this gene from map at 9, chromosome number 9. Okay. And yes, this is partial sequence of TCP1 beta, a 5' region. This is a open arrow, start 1, start codon at here at 1 and stop codon at 206. So this is when we are looking open arrow, it means it is a 5' end. Okay. So this is a partial sequence. There is a separate protein ID also, accession number for protein ID also. There is GI number. Okay. So, and the translation, translated product. Okay. So this is the amino acid sequence of that particular sequence, that particular gene, partial sequence data, CDS coding region. This is the amino acid sequence. Okay. Second, there is another gene. It started. This is a CDS. So it is a complete, not a partial sequence. This is a complete sequence for the second gene. That is, gene name is AXL2. Start from 687, end at 3158. So for looking at arrow closing and type of closing and opening of arrow, you can understand which one is 5' region and which one partial sequence, which is whole complete sequence. You will get to understand. Okay. So again, it has protein sequence. This and this is entire protein sequence. Okay. Next, this is called complement. Complement in the sense, ref 7, that a gene, the particular gene is present into the complementary strand. Okay. So when you read a particular sequence, you start from 5' to 3' region. But the gene of the DNA, if you consider entire DNA, when the transcript, replication, transcription, anything is started from the 5' origin, 5' to 3', you are going to read. And the complementary strand would be 3' to 5'. Okay. So the particular gene is present into the 3' to 5'. So that's why it is written over there complement. But your start position is 3300 and end is 4037. And this is the translated product. So that is also matter. That is also you will get to know in which strand. It is in the reference strand or it is in the complementary strand. It is present over there. You can understand. Okay. So this is the origin. It means the start of the sequence actually. From origin it has started and this is entire sequence segment. Okay. Okay, so this is a basic flat file format. In this format, you will supposed to get the data information, specifically the annotation. So for here, you will understand the distinction between the annotation and the non-annotated data. In GenBanks, where you only get the non-annotated data when it is only dumped into the SRF. When it is get to the annotated properly, it's look like this format of file it's going to represent. So now I'm just going to back and open the root or the extraction tool that is called Entrez, the retrieval system. So I've already instructed you to look back the accession number, that is U49845. Okay, you just note it down and I'm going to show you how with accession number will get the data. So just get back to the and now I'm going to type Entrez. Entrez molecular sequence database system, or it is a data retrieval tool. There is a 4 tool that is available. I already discussed. So this is the first tool you are going to utilize. So this is a browsing based retrieval system. You can use BioPython scripts to download data, but it is called the browsing based system and easier. It is GPU. You have that particular, the interface to extract the data. So I'm just going to open it. Okay, base system. So this is the web page. Just going to click on Entrez. Okay. So this is a search bar. So it is directly linked with the NCBI. Entrez is an integrating part of NCBI. So don't confuse with NCBI with Entrez. NCBI is a database. Entrez is a data retrieval tool. From NCBI, we retrieve the data. We put the query, and against the query, you will get the interpretation. So write a note on that. Entrez is a retrieval system. So now we have that accession number that we have noted from previous discussion. So I'm just going to put that accession number over there. U 4 9 8 4 5 U49845. I'm just going to click on search. See, it will show you the redirecting you to the Saccharomyces cerevisiae TCP1 beta gene partial CDS, AXL2 and REV7P genes complete CDS. So with the accession number, and that is universal. If you get over there, DDBJ and EMBL will get the same organism with same sequence. Okay. And organism Saccharomyces cerevisiae. So here you will get the FASTA. Okay. Just let me open this and I'm get back to the FASTA, how it's look like. So this is the flat file format. And we have seen into that example, the format that is given over there. See. Properly annotation is available. Okay, same flat file. So how to extract data utilizing Entrez we have performed today. Now I'm just going to go inside the FASTA. I just click on the FASTA. See, this is the format actually. Nucleotide sequence, total 5,000 onward sequence is over there. And this is the characteristics of the FASTA file. It would start a less than arrow, a close arrow with the accession number and its version. So it is a FASTA file or not the FASTA file. How could you understand that? You will see this arrow and the accession number. So this is the header, actually beginning of the FASTA file. So there is no difference between FASTA and GenBank, but this FASTA is a format, file format of that particular sequence. Okay. You can copy, you can download, you can run BLAST. So today I'm not going to show you. When we are going to cover the pairwise sequence alignment, multiple sequence alignment, then I'm going to demonstrate the BLAST and the separate types of BLAST we are going to discuss. So GenBank ID is there. This is over there into the flat file format. So in traditional way, you could directly search with the organism name, but if you have the unique identifier, that particular unique identifier, if you get that, that means the accession number, it is very much easy to identify that particular. So this is called cross-referencing. So each and everything is cross-referenced in this platform. Okay. So if you need to amplify this sequence for a particular purpose, you need to utilize in cloning sequence. You have some tools that analyze sequence segment. You have some tools you can use that. Pick primers. You can design primers for this sequence. For this particular coding region, if you need any type of targeted analysis. Okay, so today we have learned how to extract nucleotide sequence data, not any other biomolecules. We're handling nucleotide data. So next we are going to discuss what is the utility of this data. What is the utility of the first file? We are going to perform that. We're going to analyze. Next part, we are going to discuss about DDBJ, and each and every detail we are going to cover. DDBJ, I have already covered GenBank, and this is just a part one. In next part, we are going to cover different type of submission type, and we have see some extraction part, and now we are going to see some submission part also. How different types of systems are there. So we need to get an overview, and after that, we would discuss some databases like PDB and other databases that is available. As much as possible, we are going to discuss about that. And
Navamita, there are a few questions.
Okay.
You can take them and then continue again.
Okay.
In the chat.
Okay, please repeat the question, please. Would you like to read them?
One was uses of FASTA format. I'm just going through all of them together. And one is, so our gene sequence will be FASTA type and then submitted to GenBank and get accession number as completion.
No. When we're going to submit, that is in raw sequence. This is a simple flat file format and ASN.1. Actually, ASN.1. When we are going to submission, but we have ASN.1. But when we are extracting, we have FASTA.
Another last question is, when multiple sequences of the same gene in GenBank, what criteria should we follow to select the most best sequence for our study?
Most best sequence in the sense? Multiple sequence is there, maybe, but
what criteria should we follow to select the most best sequence for our study?
Okay, I understand what exactly you wanted to say. You can perform sequence similarity search, and one by one I'm going to discuss about that. Their percent similarity search. So if you have a particular segment of a particular organism, then you are going to start how much similar that particular sequence is. You can perform run blast, and there is a percent identity matrix over there. The percentage, from that particular percentage, you will understand which sequence is very similar to other organisms, like this way. And for a particular sequence, always the database contain that unique identifier. There is maybe multiple sequences are there, but the redundancy is completely removed. That is a part of processing. So there is always unique identifier. And if you need to identify, you need to identify a similar sequence or the consensus sequence or a conserved region, you need to perform blast. I think any other question I could accept?
Not at present.
Okay, so may I close the session then?
Yeah, I think you can. Okay, there is one question. Can you please outline for primer designing also from this
Yeah,
sequence in future?
I will take separate session for primer designing. Primer designing, actually we are uh, utilizing the concept of local alignment search. The main principle of search, before primer designing we have to perform the blast, and after that gradually we'll go for primers. If you need to identify particular region where exactly your primer going to be attached, for that purpose you need to perform blast, the similar sequence, and then you with the sequence similarity you will get a particular sequence. That sequence could be act as a template for your primer designing. So this is a sequential process. I'm going to discuss one by one.
Okay, I think we don't have any more questions as of today. Uh, yeah. So thank you very much again, Navomita, for an excellent and informative and engaging session, and also answering as many as questions as possible. So today's discussion took us an important step forward, which understood through the FASTA format and how it can be actually retrieved, compared, and analyzed.
Okay,
And
now okay, just interrupting once.
yeah,
I get understand when multiple sequence of the same gene is in bank, what criteria should we follow? The simpler select the most best sequence for our study. Understand there should be a searching criteria. Specifically, she has asked. Yes, for when we're looking for blast, there would be some e-values are there. E-values and percent identity, percent similarity. When we are going to perform blast, we'll see. According to that, we'll make a decision. Thank you.
yeah, I think it will make the participant more clear on her question. So yeah, so I hope today's session helped the participants to see how these concepts move beyond theory and become more practical tools for their day-to-day experiments and biological research. And I will also like to thank everyone who joined us today and continued this learning journey through this week 3. We are approaching the final week for this month, and hopefully we will continue again next month. That we will notify. But yeah, for this month we will have one final week next week. And then thank you again, Navomita, for sharing your time and knowledge with us. And thank you all the participants for being part of the session. We look forward to again on the final session on week 4 next week. Thank you.